Life. It is so complex, but what makes it so special?
We don't know for sure, but there is one special ingredient that is common to all living things.
DNA. The molecule that carries the genetic information of every single living thing.
DNA occasionally encounters mistakes in its coding, called
Mutations. And these mistakes can sometimes have devastating effects, which currently do not have conventional medical cures.
What if there was a radical way in which these mistakes could be fixed?
Well, there is now a way in which we can fix these mistakes through a process called
Gene editing, where scientists are able to alter and correct the mistakes by removing or replacing faulty genes that cause the disease. The fixed genes would then go on to produce working proteins.
Gene editing evolved from gene therapy and clearly commenced in the 1990s with the discovery of the first programmable editing technology called zinc finger nucleases. Nonetheless, this technology was not very accurate, and the process could be easily disrupted by surrounding cellular activity. Scientists then followed upon this with newer technology called TALENs. Though it was more specific in targeting than zinc finger nucleases, TALENs also had its flaws, namely relative large size which impeded its distribution.
Despite these technologies' flaws, they represented the frontiers of medical biotechnology, with their achievable ability to cure previously incurable diseases, most notably genetic diseases.
As a result, all of the unhealthy traits, which is the disease, will be completely gone and cured.
However, as pointed out in the article, gene therapy sharpens up to heal genetic diseases, published in The Age on the 15th of November 2015.
Scientists really struck gold in 2012, realizing the potential clinical applications of a process that has already been present in nature for millions of years.
It all started with the discovery of the
Strange sequences in the genes of the bacteria Escherichia coli.
It turns out that
Bacteria can get infected or sick like us too. The sequences called CRISPR act as the immune system of the bacteria. Upon first contact with an invader, bacteria produce Cas enzymes which bind to and cut out a section of the foreign DNA. This section of the DNA would be integrated into the bacterium's genome, what is called a spacer.
bacterium would use this space in the future to better detect and eliminate that particular invader, just like a vaccine does.
Scientists then used nature's deliberate genome editing system to create the most
Affordable, versatile, and precise gene technology to date. CRISPR-Cas9 is also relatively easy to use because of the easily programmable guide RNA for a range of diseases, from simple single gene diseases to complex multiple gene conditions like cancer.
The process of CRISPR is as follows. Firstly,
An easily programmable strand of guide RNA tailored to the specific faulty gene will bind to the DNA strand. 9 Cas enzymes produced by CRISPR then act as a nuclease, which cuts the DNA. The final stage would be homologous recombination, where the cut DNA is repaired either naturally by the body or artificially via insertion of a functional copy of the gene into the DNA double helix.
and the vast array of potential applications for this gene technology is staggering.
An obvious use for CRISPR would be
For somatic modification, when incurable diseases are cured permanently. This would bring all disease sufferers a chance at a normal life free from suffering.
Imagine the number of people who could benefit from this technology globally.
70,000 cystic fibrosis sufferers, 80,000 hemophilic people, 300,000 people with thalassemia, 500,000 people with Huntington's disease, and more than 2 million people with sickle cell anemia will be cured. Millions of lives could potentially be
In
saved.
addition, CRISPR cures in
Germline therapy, where changes in the genome are passed down generations.
This would offer an opportunity for all future offspring to be rid of this disease. Branching off from altering the genome of preborn babies is the idea of
Designer babies. Babies could be altered so they can possess desirable characteristics like beauty, athleticism, or intelligence.
CRISPR, through
Xenotransplantation could also be the solution to organ donor shortages.
and the practical applications of CRISPR extend well beyond the boundaries of a clinical setting.
Gene drives used to eliminate or alter population of organisms would neutralize the threat they pose to their environment or to us. For example, mosquitoes or ticks carrying disease or pests would be wiped out or modified to begin with. Even our diet could benefit from CRISPR. Through genetically modified crops, food would be enriched to be more nutritious for additional health benefits, or even modified to be more convenient for our fast-paced lifestyles.
Despite the extremely exciting and beneficial real-life applications of gene editing, we must realize that there will exist several issues, both ethical and practical, and that these issues must be resolved before further advances occur.
In specific reference to the current CRISPR technology, there are still several practical kinks which have also plagued previous technologies. When a new gene is inserted, there are problems with distributing the editing technology to the desired site. Maintaining cellular function after gene integration is also a problem. Moreover, cutting genes exposes the DNA to the risk of mutations and unwanted medical issues. The process of distributing the enzymes is also inefficient. There is currently no way to
viably deliver the Cas9 to all cells.
It must be noted that concerns regarding safety have been raised. Clinical gene editing, like all other medical procedures, have at least a small margin of failure. 100% effectiveness cannot be guaranteed 100% of the time. For example, the wrong gene might be accidentally targeted, possibly inducing harmful consequences for a patient. Unexpected side effects may also surface as a result of the procedure. For instance, suppressing an unhealthy gene may trigger unexpected activity in the surrounding genes, leading to decreased immunity to another disease.
Gene editing's numerous applications raise some considerable issues on their own.
Gene drives could have unintended effects for ecosystems. Social division because of genetic discrimination could stem from designer babies. Germline therapy could irreversibly condemn an entire line of descendants to unintentional mutations and genetic damage.
And the prospect of derailing the natural course of
Evolution is a distinct possibility of
genetically modified crops.
There is also the issue of a lack of regulation and forethought surrounding the technology and its progress. There are currently no regulations regarding any of the gene editing technologies or its applications. As a result, people could recklessly use the editing technologies, such as insufficiently trained personnel performing complex, unsupervised work on human embryos in fertility clinics. People could also exploit these loopholes by intentionally abusing these technologies, such as weaponization,
biological warfare, or terrorism.
CRISPR, the technology that has amazed and mesmerized the global community with its simplicity, its accuracy, and its versatility, is destined to radically revolutionize the medical field. Previously incurable diseases like aggressive cancers and innumerable genetic diseases can now be entirely eradicated. However, several issues are raised by both gene editing in general and the CRISPR technology, which need to be addressed before unintended consequences can spiral out of control.
Questions now are:
where will gene editing go from here? Will it be the silver bullet for treating all diseases given its volatile power? Would regulated gene editing impede its progress, and will it be a vital part in ensuring safety and its proper practice?
Genetic editing will soon leave a palpable mark upon our everyday lives. It is up to us to ensure that this will be a positive one.